Hello, everyone. Welcome to the third and final day and final session of UC GIS week. You are sitting in GIS and Aquatic Worlds. Today, to start us off, we will have Sarah McTagg from UCSD. Sarah, can you share your screen?
Yes.
Perfect.
All right. Can you guys see my slide?
Yes.
Perfect. All right. So I'll get started. Hi, all. My name is Sarah McTagg, and I am a lecturer at UC San Diego. I want to thank you for attending my talk during the GIS plus Aquatic World section of the conference titled Who's Next Door? Using GIS to understand neighbor patterns of coral and algae at Palmyra Atoll. I first want to state that I'm fully in support of the UC-wide strike for increased wages and affordable childcare occurring during this time. And with that, let's get started. So, reef ecosystems are built up by coral, which are animals that have what we call a symbiotic relationship with photosynthetic algae called zooxanthellae. The coral gives these algae a safe place to live, and the zooxanthellae perform photosynthesis to provide food and oxygen to the coral. Reef-building coral can form elaborate structures out of calcium carbonate and create shapes that look like brains, fans, and even cabbages. Corals rely on three main things for survival. The first is sunlight, because as I mentioned previously, their symbiotic algae utilize sunlight to perform photosynthesis. Next is room on the reef floor to have space to live. Lastly is water currents that carry tiny food for the coral to eat. This food is called zooplankton, which are microscopic animals that float around the ocean. Now, corals will compete against one another to access these three main resources in a process that is called resource competition. In certain situations, when corals are within the same neighboring area as one another, this can activate certain competitive behaviors that lead to intense battles between the two animals that can ultimately lead to injury or even death of those participating. So you may be thinking at this stage, what did these battles look like? Are there fistfights like something out of a West Side Story narrative happening at the Great Barrier Reef? The truth is, these coral battles are much more fascinating. There are a vast number of ways that corals can fight one another, including utilizing sweeper tentacles to cause injury to the neighboring coral, which is what you see in this video playing on the slide. Other competitive behaviors include excreting harmful mucus on one another, shading one to limit the other's access to sunlight, or excreting toxic chemicals into the water. There are many reasons why it is important that we study these interactions, but one that I am mainly focused on is how these competitive interactions impact the spatial distributions or the arrangement of coral within their environment. To understand how these types of interactions affect the spatial distributions of coral and algae, we focused this project on understanding the neighborhood compositions of these organisms. Examining these neighbor patterns will allow us to understand who typically lives near who and who typically doesn't live near who in a coral reef ecosystem. Therefore, we will gain a better understanding of how processes such as competition impact where these organisms reside within their environment. All data for this study were collected on Palmyra Atoll, which is a coral reef that is based in the Central Pacific. In September 2013, 14 sites were established on the north side and the south sides of the atoll, as shown here in this top figure. Scuba divers using cameras that are mounted on a custom frame took images of these entire sites. These raw images taken by divers were fused together to create finalized pictures that look like this. These large area images are excellent as they allow us to analyze a large area of reef, but are also incredibly detailed so that we will be able to look at the neighborhood area around each individual coral and algae in this image. After all images were traced, organisms of the same taxonomic classification were filled in with a set color. So over all those 14 sites, we traced 52 different species of coral and algae, and in total, 49,318 individual organisms. To analyze the neighborhood of each coral and algae in a site, a buffer analysis was utilized in GIS. An example cartoon here shows what each of these buffer areas look like. They are a polygon of a distance of 10 centimeters that is mapped out around each coral and algae in a site. Utilizing buffers for this study allows us to not rely on the centroids of these organisms to perform spatial analyses, but instead takes the shape of each individual organism into factor, as the centroid could poorly represent a coral or an algae, as both organisms are extremely variable in their shape and size, as you see on these examples on the right. From these buffers, we were able to determine the exact neighborhood composition of each organism. This bottom panel here shows the neighborhood buffers of one taxon, so one species, mapped out in GIS in just a subsection of a site, just so you get an idea of how many buffers would be plotted out in total. We then calculated the percent area that all organisms take up in each buffer space within a site. Using this data, we could then determine which coral and algae species are typically neighbors and which are not. So now for our results, which consist of 3 main takeaways. One is very few significant neighbor patterns were found in total. This means that most of the time, species either had random neighbors or they weren't high enough in abundance at Palmyra Atoll to cause significant patterns within our results, even if they were or were not frequently neighbors with a certain species. Second, most of our significant neighbor patterns were positive, which meant a pair of species were neighbors frequently. These mostly were intraspecific, which this word just means organisms of the same species are most likely to be neighbors at Palmyra Atoll. 3 of those significant patterns that were negative, which meant the organisms were not neighbors frequently, these were mostly interspecific, which means organisms of different species were most likely to avoid one another. We can now see how competition between corals definitely impacts the spatial distribution of these organisms in their environment. Coral defense mechanisms, such as those sweeper tentacles and toxic chemicals that I discussed earlier, can both push away baby corals that have settled nearby and damage established adults within reach, which protects the coral and maintains space around their neighboring area. Therefore, these intense fights lead to different species typically not neighboring one another, which is now apparent in the results of this project. But why then do we see corals of the same species neighboring one another quite frequently? Corals of the same species do not fight with one another as frequently when compared to those of differing species. Conspecifics that settle next to one another will most likely either stop growing in the region of contact or actually fuse their adjacent tissue together, which is what is being shown in panel E of this figure from Forsman et al. 2015. Therefore, due to this lack of aggressive behavior between corals of the same species, that is why there are so many positive intraspecific patterns in our results. However, there are other processes on the reef that are affecting our results as well. We also know that some species of coral perform an asexual reproductive act called fragmentation, which is a coral's ability to split into multiple viable corals from things such as storm events and waves, and is what you see in this top image here. The process of fragmentation leads to heavily monospecific spatial patterns, which just means a lot of similar species neighboring one another, also contributing to our intraspecific neighbors in our results. Also, by utilizing the diverse structure of the reef environment, certain coral live in specific locations of the reef, not inhabited by others. Examples include fungia, which is an unattached coral that lives solitary on the sandy bottom of the coral reef floor, and encrusting Paredes superfusa, which lives in very cryptic locations, such as in crevices of rocks within the substrate, where bigger corals, such as brain corals, cannot settle due to their size. Because these corals live in areas where others do not reside, they frequently only neighbor conspecifics, so those of the same species, and nobody else. This further leads to positive intraspecific patterns and negative interspecific neighbor patterns in our results. The last thing I will touch upon are those results that involve algae. So the only species in our results that does not neighbor itself is an algae called Halimeda, which is what you see in this image here on the left. Halimeda does not neighbor itself due to a really cool process called biotic self-organization, which is the formation of patterns in the natural environment via biological or physical properties. Here you can see imagery of mussels in a laboratory setting, going from randomly distributed to performing this spatial organization in a time period of 24 hours. If you take a look at this imagery on the slide, these green polygons are Halimeda from 4 different sites at Palmyra Atoll, including a subset of one site in panel E. The spatial pattern you see being displayed in these sites is due to Halimeda performing biotic self-organization. This is why there are almost these honeycombed shaped patterns, which I have now highlighted with red lines. These patterns lead to large gaps of Halimeda colonization from some sections of the reef floor. Due to this formation, Halimeda are also found in lines with few individuals of the algae neighboring one another. Therefore, because of this self-organization, Halimeda is found to not neighbor itself within our results of this study. And now for conclusion. So in this study, we developed a methodology that allowed us to examine the exact neighbor compositions of all coral and algae at 14 sites of Palmyra Atoll. This method we developed did not utilize centroids to do spatial analyses, but instead used the distinct shapes of the organisms. Our utilization of GIS programming and large area imagery allowed us to do this. Next, in our results, we found few significant neighbor patterns. Of those that were significant, most were positive and intraspecific due to the little competition occurring between corals of the same species, as well as processes such as fragmentation. On the other hand, corals of differing species did not neighbor one another due to intense competition for resources and due to some corals living in particular parts of their environment where other species are not found. And with that, thank you for listening, and I am now opening the floor for any questions.
What a cool presentation. I didn't know
Thanks.
corals could be so aggressive.
Yeah.
Does anybody have any questions for our presenter? I actually I would love to hear a bit more about like the symbiotic relationships of corals. Like how does like grouping together in that shape beneficial to them?
Yeah, so you were talking about those green types of blobs. Yeah, so
Yeah.
that's this cool plant called Halimeda, which they grow in these weird patterns where they'll jut out at specifically 45-degree angles. And there's actually still a lot of work out there to why this is. Basically, we're kind of figuring out, is there something with how they capture sunlight? Is there something with how they grow on the reef floor? So that's actually still up for debate. But we do see these patterns in the reef environment quite a lot when that plant is present, which is pretty cool.
And then when like corals fuse together, is that how does that like do they work as like one organism?
Yeah, so then technically, as they combine their tissue and fuse their skeletons together, because coral are kind of this colonial organism, so they have this giant skeleton with a bunch of what we call polyps growing off. And those polyps all are kind of separate entities in a way where they can capture food and capture sunlight. So when the skeletons of these two kind of masses of coral join together, basically things are still running the same. All those little polyps are still doing their same kind of thing where they're collecting energy via sun or collecting food, but now they're just kind of a bigger structure at that stage. So yeah, pretty cool stuff.
That is super cool. Sorry, you can clearly tell I'm a human geographer. Where I'm like, oh my gosh, the
Things
ocean.
fused together. Yeah. A little bit different than
Awesome.
people.
Yeah. Does any of our audience have any questions? I will stop taking up all the question time.
Just kind of have a quick question for Sarah.
Mhm.
How did like GIS, at what component did GIS start making to your study of like corals and marine biology? Was it something like you were kind of curious of, or was it like, did someone bring it to your attention, or did it like naturally, did you think like, oh, like I should learn this. I think they could overlap well.
Yeah, actually, my advisor for my master's was talking about running this neighborhood type study and interested in how things kind of revolve around each of the corals within a really small space, so that 10 centimeters. And that's when it kind of popped into my head. There's a tool in GIS that is so easy, the buffer tool, to do this process. So based on stuff we were talking about, neighbor patterns, and then I have a background in GIS previously as an undergrad, that I was like, let's just stick this all together and make a project out of it. So that's kind of how it formed.
Oh my gosh. Thank you so much, Sarah.
Thank you.
Zoom claps, everyone. Next up, we have Paige Hoel from UCLA. Paige, would you like to share your screen?
Yes, definitely. All right, so I'm going to show you guys a story map today. I'm going to put the link in the chat if you'd prefer to just roll through on your own, but I'll take us through a little tour here. Yeah, well, first of all, thank you all so much for coming. Thank you to the organizers for getting this wonderful like accumulation talk together. Very exciting. Yeah, and I'd like to also show my support. I am a TA as a part of the UC system, and I'm in full support of the strike and everyone involved in those efforts. But today I'm here and I'm very excited to be talking about a title my talk, which is titled The Human Kelp Connection. And I'm going to explore if we can use GIS to find any connections between anthropogenic influence and kelp patterns in the Southern California Bight. So I'm going to start us all off though with no GIS. I'm just going to start us off on a hot summer day, actually. You are scorching on the beach. It is mid-July. It's crowded. You're ready to just get out there you dive into the waves. And if you're in Southern or Central or Northern California, it's a little bit chilly at first. You know, you paddle out, you make it past the breakers, and you finally get out there and the sun's on your skin and the water's cool and you start to relax. But then suddenly you feel a tickle on your foot and you're like, you jolt up and you think to yourself, oh my God, what was that? And you look down and there's like a very, very, very high chance that this is what you see. You see Macrocystis pephra, which is giant kelp, which is an absolutely iconic species of kelp. And it really plays into so much of the culture that we have around beaches in Southern California and the rest of the California current system. But especially in the Southern California Bight, I think anyone would be hard pressed to think of a beach day where they haven't seen this on the beach or they haven't touched a piece of kelp. And I've always been fascinated with kelp. I grew up in Southern California and did my undergrad in Santa Barbara, and there was this massive, beautiful kelp forest just offshore. And I was curious. I studied physical geography and physical oceanography, but I was kind of curious, like, why is kelp so successful here? It seems like kelp do a relatively good job of growing. And this is not just my opinion. It's actually interesting in terms of giant kelp worldwide. Giant kelp grows in so many different ocean ecosystems globally, but especially in the Southern California Bight, it is among some of the healthiest in the world. And this is an image I have. I took this off of Santa Cruz Island in the Channel Islands. And you can see like the kayakers can barely even get through it. Like you're just like pedaling through mud. It's really beautiful and so cool. So why is it that this species is so successful here in Southern California? There's actually kind of 4 key reasons that I can use to compare it against other giant kelp ecosystems. One is that in the Southern California Bight specifically, going from Point Conception down to Baja, Mexico, is that there is protection from storms and large wave events. Part of this is the geography of the Southern California coastline, kind of at that awkward right angle. And that actual, that modification allows for waves to be buffered a little bit. The other reason why waves are buffered in the Southern California Bight is the actual Channel Islands. They provide a physical blockage of large swells. So although there are large wave events in Southern California, they are not as frequent and as dramatic as they are in Central and Northern California, where the coastline is just directly exposed. Another thing that's going through the California Bight is the lack of predation. And what I mean by that is there are very few predators on giant kelp. So in Central, Northern California, and the rest of the California current system, they've experienced some very massive die-off events due to purple urchins. And thankfully in Southern California, purple urchins are not an issue. So there's relatively little predation on giant kelp in Southern California. Another reason giant kelp does well in the whole California current system is the temperatures. It's a Goldilocks situation. It's just right. It's not too hot, it's not too cold, but it's a little bit colder, and kelp loves that. So optimal temperatures for growth for giant kelp, also a winning factor. And the last one that I want to bring up is a very ample supply of nutrients. Now, if anyone in here is an oceanographer or in the earth science, marine bio world, you know that upwelling is a huge influence on the Southern California and the whole California current system. But just to give a really brief overview of upwelling, upwelling is when cold, nutrient-rich water comes up to the surface. And so what I'm showing you here are just a couple of like standard profiles from the ocean. It shows that with depth, water gets colder, but also it's richer in nitrate. And nitrate is a primary nutrient for phytoplankton, micro and macro plankton is a better way to put it, micro and macro. So yeah, when we're talking about the California current system, we have a wonderful upwelling mechanism that brings up this cold, nutrient-rich water to the surface. And that is awesome for kelp. The kelp love it. And kelp needs a relatively consistent supply of nutrients. Their natural storage is relatively low. They will run out of dissolved inorganic nitrogen after about 2 weeks. So they need consistent supply of nutrients, which is commonly found here in Southern California. Here's another wonderful image. This is from an artist, Frankie Grant. You see this little Garibaldi down in the left-hand corner, just so wonderful. So I'm telling you all these things about how kelp is so healthy here and it's doing such a great job. But that's not to say it's invincible. And kelp has been disturbed in the Southern California Bight. But when was that and why was that the case? So I'm going to start by showing you 2 maps. So on the left-hand side here, these are locations of anywhere kelp has grown between the years of 1988 and 2021. This is using a data set from LCER at UC Santa Barbara, and they use Landsat imagery to track locations and size of kelp farms. I'm sorry, kelp forests, not farms. And then on our right-hand side, this was the standing stock of kelp in winter of 2015. Winter of 2015.
You might understand that, or might remember that in winter of 2015, there were things going on in the California current system, namely a marine heat wave. So what the marine heat wave did was it raised the temperature of the entire Southern California Bight. First of all, when the temperature is raised, it's less ideal for growth for giant kelp. Giant kelp will get a little bit colder. But with this increase in temperature, what happened was it stratified the water column. And when the water column was stratified, suddenly this upwelling of cold, nutrient-rich water was suppressed. And the surface of the ocean was not receiving as much nutrients previously. What I wanted to see was during this marine heat wave that kind of devastated a lot of kelp forests in Southern California, what was allowing the kelp forests that did grow or that recovered quickly, what helped them do this? Another figure I want to show you to just talk about this marine heat wave is this one right here. This is a time series. So on the top, what we have on our y-axis is just the actual extent of the kelp forest. And on our x-axis are years. Now, there are actual kelp locations. These are drawn by the California Department of Fish and Wildlife. There's a whole history as to what these locations actually represent. But I can tell you that location number 3 is off the coast of San Diego. This is off the coast of Point Loma. And what you see is that kelp, depending on its location, is very ephemeral. It can grow, it can die, it can grow, and it can die. Very resilient. But what you see here is a massive die-off between 2015 and 2017 during this marine heat wave. On this location underneath, location number 13, which is off the coast of Palos Verdes, further north in Los Angeles County, you see that during the marine heat wave, there actually is not a massive die-off event. And you would be hard-pressed to detect that anything even happened, comparing this to the rest of the running mean. So why was this? Why is this kelp in Palos Verdes doing so much better than this kelp in Point Loma during this marine heat wave event? And my previous research in my PhD was actually studying wastewater outfalls and anthropogenic nutrients from wastewater outfalls. And I can tell you that there is a massive wastewater outfall that pumps a bunch of dissolved inorganic nitrogen into the ocean at Palos Verdes. So I came up with the question looking at this data, well, could our anthropogenic nutrients, either from wastewater outfalls or from river inputs, could they help support kelp through that marine heat wave? Were they supplying dissolved inorganic nutrients to these areas that were not getting a regular supply from upwelling as they were previously or during normal conditions? So that's my question, and the answer is yes. It's actually pretty cool to see how some of this data comes out. So I have a bivariate color scheme here. This is a lot of colors. I'm going to start by talking about the blue on the bottom. So the blue is a normalized marine heat wave area. Essentially what this is taking the average area of each of these little 2-kilometer grid cells, the average area of that kelp forest during the marine heat wave and dividing it by what the average area was before the marine heat wave. So a bright blue would represent a completely its normal area, or actually went above slightly its normal area during the marine heat wave. And a white would be that it did not maintain its area during the marine heat wave. On the y-axis in the pinks, this is talking about, this is a metric I created. I call it influence days, but these are the number of days in the year in which the actual water column at this location goes above that nutrient threshold that kelp needs to grow. But what's interesting about this metric is that this nutrient threshold, I'm only looking at anthropogenic nutrients. And so I'm able to calculate, okay, throughout the year, at some locations, there's more than 10% of the year, anthropogenic nutrients can supply enough alone to help kelp grow. And so you see then in areas that are purple, they sustained their area through the marine heat wave, and they had a high influence from anthropogenic nutrients. And no surprise to me, as having looked through this data a bunch before, not in the lens of kelp, but just in the lens of nutrients alone, that we see some big hotspots, a big hotspot around Palos Verdes. We see hotspots around northern Orange County, and some around southern Orange County and northern San Diego County. So my thought is yes, that I do think anthropogenic nutrients are can help to sustain kelp through some dramatic events such as a marine heat wave. I want to briefly mention, why am I even talking about this at a GIS conference? Like, what have I done that's so special to bring it here? And my short answer is I haven't really done anything special. This is just a pretty basic spatial analysis. But what is kind of, I think, unique about this work is that I'm putting together a lot of different types of data that really mesh well into GIS. So the kelp stalks and the kelp forest area, those are all rasters. Those are all Landsat imagery that you can play with and, poke around at. But the actual ocean state, we're using a very advanced coupled physical chemical model that we run at UCLA that's 4-dimensional. It comes out in NetCDF. By putting these two together, though, we get a really good idea of what the actual chemistry of the ocean is doing, and then studying kelp on top of that. A lot of previous kelp studies, they only use sea surface temperature from satellites to estimate the amount of nutrients. They don't take into account deep wastewater outfalls and other anthropogenic nutrient sources. They're just using the sea surface temperature to estimate nitrate, which I think is definitely wrong. There's big room for improvement there. My other question that's related is, after the marine heat wave, did areas with anthropogenic nutrients recover quicker? And again, my answer is yes. After the marine heat wave, areas that receive large amounts of anthropogenic nutrients also bounce back quicker. Again, we see these hotspots around Palos Verdes in Orange County, and then where they're not as influenced in Santa Barbara, doesn't make a large change. So I'm going to leave you guys with this. I just wanted to show, just as an example though, how lame this data looks, not in GIS, not in a map. They just have these like stale box plots that like don't even tell a story. And it's just so hard. When you partition it by county, it makes no sense. But then I can put a figure like this into my manuscript and things make a lot more sense. So just advocating for more use of GIS within the marine science world and literature. So that's all I had. I'd like to thank my collaborators at SWRP, the Southern California Water Resource Protection Project, and UCLA.
Thank you so much, Paige. That was incredible. Yeah, boo box plots. Did we lose Paige? Maybe. Oh, hello. Sorry, you froze for a minute.
My internet right now is deciding to be funny.
Thank you. So we have a question from Christy in the chat. Is the kelp bloom off North OC close to the Seal Huntington Beach because there's a wastewater pipe outflow close to shore there that caused a headache for some environmental engineers I knew at Caltrans. So I
Yes.
guess is that kelp bloom close to that beach?
Yes, ma'am.
Now you know.
Yep, it's pretty cool. So our yeah, you can see right here.
Yeah, that's the one.
Yep. It's pretty amazing. So the model that we're using so this is a model developed at UCLA and at SCWRP. It takes into account large POTWs, which are like the large I forget point outfall treated wastewater. So the large pipes. So there's 4 massive pipes in Southern California that pump out about 1 billion gallons a day of treated wastewater. There's one off the coast of Palos Verdes. There's one in Orange County. There's one in Santa Monica. There's one in Point Loma. Now, the small POTWs, which is what this is a result of, I think, they're closer to the surface. So the large wastewater outflows, they're beneath the surface. They put them at about 60 to 80 meters in hope that the dissolved inorganic nutrients get downwelled and they don't interact with the surface in the euphotic zone. That I don't think is the case. But especially with the small POTWs, those are way closer to the surface and they're going to interact way more with the kelp. They're just in the coastal zone. And so that's cool that you point that out because yeah, I agree. I think there's a lot of little sources of nutrients, but a little source of nutrients could really help sustain kelp. In this marine heat wave, so much nutrients were repressed from the actual euphotic zone where they need it. So a little bit would go a long way. So I think, yeah, that could definitely be a result of that.
Yeah, there's like nothing towards Laguna.
Yeah, so true.
Yeah.
So
Because there
true.
used to be the creek that would outflow from Laguna Beach's main beach, but then they like dammed it up. So it's been an issue about undamming the creek to deal with the loss of sand down south. So it's interesting to see that like even down south, there's like some, but definitely like damming that little creek. Like this was like decades ago.
Mhm.
You can see the impact down there.
Yeah, you can definitely still see the influence. Yeah.
Anyway, those are just my 2 cents.
That's
Good job,
awesome.
Paige.
Yeah, thanks, Christy.
I also have a question if we have time. Awesome. So, yeah, my strong background is not in physical oceanography. So if this is a little bit off, but given that you are you taking into consideration, because you're just talking about the depth at which the nutrients are coming out, is there any impact as well with, say, like water currents or such? Is that impacting where the nutrients drift, like with the California
Yes.
current going southern? Because I know Point Loma and Tijuana, like ton of stuff coming out in terms of nutrients, but it's so interesting the kelp did downfall during that marine heat wave. So I was wondering if it's somehow the California current had.
Yeah, that's a really great question. So for example, here for looking at Palos Verdes, the outfall, I should actually have a symbol on this map, but the outfall is like it's just out. Can you guys see the cursor? Is that on? It's probably not on the screen. But the outfall is, if it was distributing evenly, it would be going throughout the whole peninsula. It would be going around the Palos Verdes Peninsula. But because the current is predominantly southward and southeast, it pushes all the nutrients down and towards Long Beach. And so that's why we don't see a wider distribution. And in general, pushing them further north towards Malibu, for example, it is such a massive amount of nutrients that comes out here. But the model is a coupled physical biogeochemical model. So it in a very advanced way takes into account all of the currents. And specifically our model's resolution is like 300 meters, which is really good for resolving. Yeah, it resolves like the sub-mesoscale, which is like you can resolve all the little currents going in and around the shore. So that's a great question.
Cool. Oh, that's awesome.
About Tijuana, I think so. Around Palos Verdes, first of all, that there is a wastewater outflow, but it's super deep. And second of all, it does advect offshore. The currents get pushed offshore. That we do not know how much nutrients are coming out of Mexico and out of Tijuana.
Yeah.
Untreated wastewater that we're trying to make good estimates for, but that is to be updated. Still, it's not enough to impact. I don't think it goes far enough north to impact the kelp populations that are around Point Loma.
Got it. Awesome job. Well, thank you for answering my question.
Yeah, thanks, Sarah. I appreciate it.
So it seems that our third presenter is unable to join us today. So I just wanted to open back up the floor. Sarah, if it's okay with you and Paige, if anybody has any questions for both of our presenters. I personally could just listen to you talk, both of you talk all day. So I'm just happy if you guys want to chat between each other. This is really interesting. I actually have another question for Paige. Have you gotten a lot of like positive response to basically mapping this sort of data out, or like why hasn't it been done previously?
Okay, Alexis, this question burns in my brain all day and all night. It actually shocks me to my core. So I just got started on this work in earnest in like June. So I've always been interested in like ways of applying our physical biogeochemical model that we run at UCLA. It's a really interesting model, but it's like very intense and technical, and it's the data is not distributed widely to the public. So that's been in my brain of like, how to make this more palatable for people that are not physical oceanographers. On the other hand, I've also been fascinated with kelp. UC Santa Barbara, LTER, they've done so much kelp research over the past 30 years. It's insane. And so they have this very rich data set where it's all Landsat imagery going over the Southern California Bight seasonally. So there's 4 records in a year. It's a lot of really great data. And I do not know why there hasn't been done with it, to be quite frank. I think there's still this weird, and Sarah, I'm curious to hear your opinion. I think there's a weird lack of GIS in like application aquatic systems. I think part of that is the intimidation of depth. Thinking of like, how can I compare this to land? Why are there so many wonderful ecology studies with GIS? There's no depth component. So I think that has intimidated people in the past. And then just like industry, like everyone uses whatever software they use to analyze things. But I think it's crazy. Like the ocean is everything is so spatially connected. I don't know why more GIS stuff. So that's a great question, Alexis. I don't know. I'm curious to hear your opinion, Sarah, just on GIS and the aquatic world in general.
Yeah, I think it's also kind of a mix of things. One, that land obviously has been studied a lot more than the ocean in terms of like, there's just so much we don't know about the ocean. And I feel like lacking that knowledge kind of leads into us lacking the tools in order to gain that knowledge in a way, if that makes sense. And everything Paige said, spot on for what I've experienced. I think also oceanography in its own way is kind of tuned out to taking in new ways of analyzing data beyond just like, we're going to do MATLAB, which is just a coding program that tons of like physical oceanographers especially utilize. And I've had conversations about, oh, why would we use GIS? We don't have to code to do anything in it. And it's very much this kind of prestigious way of looking at things and saying that, okay, if it's too easy to use, that means it's not producing worthy science in a way. And I'm trying in like, especially I'm a lecturer for Intro to GIS at UCSD, and I'm trying to push the narrative that like, just because something's easier to use doesn't mean it's not worthy of using. Like it's even better because so many more people can utilize that program. So I think that's my main 2 points about that.
I have a question from Amy in the chat. What do both of you see are next in terms of the steps in your research, both in understanding more about your topics, but also in regards to affecting policies around both corals and kelp?
Yeah, that's a great question. I'll go first just since I was unmuted anyways. So I finished up my master's actually about 2 months ago. So my research on this is at this point just publishing this work. I see myself in the future working with GIS in terms of analyzing environmental based work, and not necessarily coral reefs, although it's just such a niche topic that sometimes it can be hard to branch out in terms of beyond academia, given that I don't have my PhD. In regards to affecting policies around corals, a lot of my research is more just knowing on the straight ecology side of how these organisms work. But in terms of like a huge thing right now is coral planting for like coral reef farms in order to kind of grow up the population. So we don't have, no coral reefs basically in the future. So that's where these coral reefs really come in handy. And a lot that we can learn from stuff like, how do these corals fight with one another and distribute themselves in their environment? I think understanding those types of distributions allows us to move forward in creating these new reefs that are man-made in a way. Obviously, we need to know how things are interacting because if we put two together and it's not going to work out, obviously we don't want to put our money into a project such as this. So that is one way that understanding the spatial distribution of coral could really come in handy in the future.
Yeah, this is a really cool question. So I'm currently this is kind of the second chapter of my PhD, and then my third, I'll have a little bit more liberty. And what I really want to now explore is making like a quantitative estimate as to how much kelp is serving the Southern California bight. So I can take a couple different angles on this. First of all, just making an estimate as to how much dissolved inorganic nitrogen is kelp taking in. So when there is an excess of dissolved inorganic nitrogen, in case anyone here isn't from an aquatic background, a process happens called eutrophication, where anthropogenic nutrients then disturb the ecosystem. You end up with large phytoplankton blooms because they're taking advantage of all these excess nutrients, and it can really deteriorate the water column. And it's a large concern for coastal communities. You can end up with massive dead zones where fish can't live, and a whole other like cascading effect. But kelp can draw in nutrients. Like, it is a powerhouse. If you give it enough nutrients, it will grow fast. And so I'm curious as to how much kelp has done in terms of buffering these dissolved inorganic like nutrients, specifically nitrogen, in the Southern California bight. So that's one task that I'd be interested to do. And I can do that because I have a coupled physical and chemical model. And then we have all these estimates of how much kelp has actually grown in proximity to all these releases of anthropogenic nutrients. I think I could make some good estimations as to how much kelp has helped buffer. That being said, then with something like GIS, I think it'd also be cool to look into suitability mapping. So kelp is hot right now. Kelp is super hot in the world of like oceanography, and it's hot for a couple reasons. One, as a carbon offset mechanism. Companies are interested. We may have kelp farms and we can buy kelp and then sink the kelp, and then then we're negating our carbon footprint. So kelp is really hot. Kelp farming is really hot. It's also, interest in Southern California again to mediate anthropogenic nutrients and also to help buffer ocean acidification around shellfish farms. So that's another thing too with our like physical chemical model. Is I was thinking about parameters that kind of roughly simulate the growth of kelp in a kelp forest, and to be able to like make a model estimation of, oh, what if we put in a kelp farm this large around the shellfishery, how much could it help? How much could it remediate ocean acidification, opposed to doing like an in situ experiment where you actually have to grow the kelp and take the estimates, using a model to kind of stimulate that. So those are like, you know, two examples. I could see this going in so many different directions, but those are two that are kind of hot on my mind right now.
Do you have like a dream kelp map? Like if you could just like make a map of the kelp, like what would you want it to show?
I would want it to show, I would be very curious to see, with a little bit of human intervention, how much kelp we could grow in terms of like actual kelp farms that we could farm and like use as either fuel or like a food source. So I think it'd be cool to show where should we put kelp, because kelp grows easily, especially in Southern California Bight. It's happy to grow, but there are some areas that aren't so suitable just because like there's enough turbulence from the waves that it'll get disturbed. And but yeah, my ideal map would show like, okay, here's where we should make sure kelp is growing naturally. Like here's where we should just like have MPAs, marine protected areas, to make sure the natural kelp is doing its thing. But then like, here's where we could also put farms and really like let kelp kind of buffer our human impact and potentially serve us to be sources or carbon offsetting. But yeah, I'm all on the kelp train. I'm drinking the Kool-Aid. I'm buying the hype. I think it's so cool.
Sarah, do you have a dream coral map, something that you would love to see?
Oh my goodness. My brain goes automatically so many different directions. Yeah, I think because the way in which we currently analyze corals and how they interact with one another is extremely time consuming, to go through and, you know, draw out every single coral's like outline on a reef, and do that times like 100 islands of data. It just takes a really long time. So I would love to see a compiled, you know, across the board, of different islands that we study, all of those coral already mapped out, every single individual, and we could like go to our heart's content with all the spatial stats that we could want. So that would be my optimum dream, probably.
Thank you both so much. And thank you for answering my dream map question. That's my new favorite question. Thank you everybody for coming to UC GIS Week. I'm so happy we got to end on such a good session. Can I get one more round of applause for both Paige and Sarah? Thank you, everyone. Make sure you join us all on Slack for post UC GIS Week discussion. And if you're interested in being involved for next year, please reach out to us. We love new people.
We totally love new people. Also, we have been sending out surveys in the general channel at the end of each day. So if you have a chance, please go fill that out. We'll also send it out to anyone who registered for the event. And this really just helps us figure out how things are going, any changes we want to make for the next year. So thank you so much for joining us this year on behalf of the UC GIS Week Committee and the University of California GIS leadership team.
Hi, Amy. I just saw, yeah, I just saw your message. Yeah, we could talk about it, whatever. Oh, I think you're muted.
Amy, you're muted.
I still am, even though I hit the button. I'm happy to follow up with that whenever, but I was just, you know, like curious, right? Because I think in talking with Scott and talking with others of like these migrating corals, and so, you know, like mapping them out or being able to map those species, and it's wondering like, you know, like how quickly do corals move or like their neighborhoods change, and so like at what, how often does
Yeah.
that map need to be updated, or right? And it made me think,
Yeah.
well, maybe is it like meandering rivers, right? Where like rivers change and after a while, but I don't know.
Yeah, I think I can answer it really quickly in the way that, like, for right now, we would, because we visit each of these islands every year that we collect data through Sandin Lab at Scripps. And it just takes so long to process the data that we don't have all the data actually updated to where we can use it to run these analyses. But I think because of things like storm events, they can really like take out an entire reef in a very short amount of time. So even on a year scale cannot be enough to see like the real dynamics of what's going on in terms of like resource competition and such. So that's when, like, if you go, I told Alexis about this in the comments, but if you go on YouTube, you can see people make videos all the time of these corals fighting each other, because labs do more actual tank experiments where they put them in and see how they're interacting and how fast that actual process is. It's not fast. Usually they time lapse it because it takes days for them to actually reach each other. So it's actually pretty slow in terms of how they move. But yeah, my goal would be every year to have something, but
Okay.
obviously, given how long it takes to process data, it's a bit of a struggle. So
Well, that's today. Give it 5 years, right? And
yeah, see what
press a
happens.
button and things will
AI,
be,
yeah, that would be sick. Yeah.
No, cool. Thanks for that, Sarah.
Yeah, no problem. Well, thanks for having me. Appreciate it.
Yeah, thanks, Angeline.
Oh, sorry. Sorry,
Bye.
somebody else. Yeah, bye.
Thanks all.
Have a good day, guys.